Unit 9 Inheritance Biology. One concept of genes. Mendel, while conducting his experiment, the results proposed a few hereditary factors that controls different traits of an organisms. We now call these hereditary factors as genes. Gene is a portion of DNA that resides on a locus of a chromosomes and carry hereditary informations. Alleles. As introduced earlier, genes can exist in different forms, which we now call alleles. The alleles can be defined as the member of the same gene pair occupying same locus on the chromosomes, each of which affecting a character somewhat different from the other. In general, a gene pair in a diploid organism contains only two alleles at a time, one on each chromosome. If an organism contains two identical alleles, this is called homozygous, while if the alleles differs, this is called as heterozygous. At a time, one allele is always associated with a dominant phenotype known as a wild type allele. On the other hand, the allele which is associated with the recessive phenotype is known as a mutant type allele. For example, the coat color in a type mouse is determined by a gene that has two alleles designed designated as capital B and small b. Capital B is a dominant allele which inhibits a black coat color, while small b is a recessive allele which exhibit a brown coat color. Multiple allele. When a set of more than two alleles occupies some locus on the homologous chromosomes and affects the phenotypic expressions, they are called multiple alleles. They have arisen as a result of a mutation in the gene. In diploid organisms, any two of the multiple alleles are found at a time, and these are found to influence only one character. The first case of multiple alleles was found in human blood group antigen system, which has been discovered by Landsteiner. It was named as ABO by Brainstein, who discovered that the genes has three alleles, uh, 1A, IA, IB, and IO. I stands for agglutinogen. When the ABO locus was mapped on the chromosome 9 in the human genome, it was found that among these alleles, IA and IB alleles are co-dominant to each other, whereas IO is recessive. Thus, four different blood groups A, B, AB, and O are determined by different types of genotypes. ABO locus produces an enzyme, glycosyl transferase, which mediates the synthesis of polysaccharide components of RBC membrane glycoprotein. The IA allele codes for the enzymes alpha N acetyl galactosyl transferase converts H antigen to antigen, whereas IB alleles codes for alpha D galactosyl transferase converts H antigen to B antigen. IO is a null allele which does code for any enzymes and cannot convert H antigen. Genotype of ABO blood group. Phenotype A, genotype IA, 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 and I0. Phenotype B, IB, 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 and IO. Phenotype AB, IA and IB, and phenotype O, IO. The multiple allele are also observed in the case of coat color inheritance of a rabbit. Four different coat color alleles are found in the rabbit, such as capital C, agouti, small c, ch, chinchilla, ch, Himalayan, and small c, albino. The dominancy of these alleles B follows capital C greater than capital C small ch greater than capital C small h and greater than small c. Genotype of rabbit depending on coat color. Albino capital C capital C capital C capital C ch capital C h and capital C small c. Chinchilla capital C ch capital C ch capital C small ch and capital C small h And third, capital C, CH, and small c. Himalayan, capital CH, capital CH, comma, capital C small h and C small c. Albino, small c small c. Pseudo alleles. The pseudo alleles are two different genes that have similar functions, structurally different, and are genetically linked as they are located very close to each other. Some authors have suggested that the pseudo alleles are formed due to a gene duplication and have many evolutionary roles. The pseudo alleles have many characteristic features as they occupy the complex locus, which can be divided into subloci. They exhibit the cis-trans effect. The low frequency recombination is observed in these pseudoals. Figure the pseudoals and their arrangement in Drosophila determines specific phenotypes. The Lozingi locus in Drosophila is a well-known example of pseudoalles. The pseudoalls such as IZ1 and IZ2 can exhibit cis-trans effect. The trans heterozygote exhibits mutant phenotype, the glossy smooth eye, while the cis heterozygote exhibits wild type phenotype, normal eye. Complementation test. The ability of two recessive mutation to restore the wild type phenotype either partially or completely in trans heterozygote position is known as a complementation. The introduction of two recessive mutation simultaneously into the same cell but on different chromosomes to determine whether they are alleles of same genes, that is whether they affects the same genetic function, is known as a complementation test. If the mutations are allelic, the genotype will exhibit a mutant phenotype that is non-complementary, whereas if they are non-allelic, it will exhibit the wild type phenotype that is complementary to each other. Cis test. The two mutation presence in the cis or coupling configuration on the same chromosome is known as a cis heterozygote. The cis heterozygote may have the wild type phenotype. If the cis heterozygote have mutant phenotype, the trans test cannot be used to determine whether the two mutations are the same gene. Thus, the trans test can never be used with the dominant mutations. Trans test. This is often known as a complementation test. Here, the two mutations are found in a repulsion configuration. If the trans heterozygote has the mutant phenotype, then the two mutations are the same unit of functions or the same genes. If the trans heterozygote has the wild type phenotype, then two mutations are in the two different units of functions or two different genes. In this case, the two mutations are said to be complement each other. Limitations. The complementation test have the following limitation: 1. Dominant or co-dominant mutations. 2. Intragenic complementation. 3. Polar mutations. And 4. cis-acting gene. The complementation map represents a complementation group as a discrete non-overlapping line which can be constructed by performing pairwise complementation analysis of several mutants. Suppose in Drosophila mutant A, B, C, D, E, F, and G all have same phenotype. The absence of red pigment in the eye. The pairwise combination in complementation test, the following results were produced, where plus means the complementation, minus means no complementations. From the above results, the number of genes may be analyzed as follows. 1. Here, capital A complements with B, C, E, and G. Therefore, B, C, E, and G are in separate cystone. On the other hand, F and D are non-complement with A. Thus, they are on the A cystonic group. 2. Capital B is a non-complement with the capital G. Therefore, capital B and capital G are in the group or in another group. That is in same cystonic group. 3. Cis C is a non-complement with the capital A, so it is in the other group, that is capital C and capital A is in the same cystonic group. Therefore there are three genes present: A, D and F are in one gene, B and G is in another gene, and C and E in another gene. The factor that controls trait is called answer gene. The following forms of gene are called alleles. Mutations at two different loci of same gene X results in altered functions. These two mutated versions of the gene X is called alleles. And 2. Mendelian principle. Inherent biology.